Buenas tardes. Good afternoon. We will continue in this symposium. With this symposium, we have a lot of delay. First of all, I would like to thank the organizing committee, the opportunity to be here, and also I would like to thank everybody to remain in the room in spite of the sunny day outside. So we try to recover some part of the delay, and we start directly with the session dealing with the metastatic and non-small cell lung cancer without driver mutation. That means the majority, still the majority of our patients.
So it's a great pleasure for me to chair this session with my co-chair, Dr. Masuti. My name is Yair Bar from Sheba Medical Center in Israel. And it's a huge pleasure for me to introduce the first speaker, who doesn't really need introduction, but Dr. Solange Peters, the past president of ESMO, a leader of ETOP, and the head of medical oncology at Lausanne Hospital University in Switzerland. Please, Solange.
Thanks so much. So I was supposed to complete the picture that Martin Reck was starting to build, speaking about high level of PD-L1. He will do it later on. So we'll start with the negative side of it, the negative PD-L1 patients, questioning in this patient population what might be a better option, combination, and basically, of course, focusing on CTLA-4. And we'll extend a little bit about maybe subgroups of PD-L1 negatives or very rapidly features justifying the CTLA-4 insight, thought, or even perspective. This is my disclosure slide. I will try not to cough. Sorry for that if I do, but it can happen in the middle. So basically, we speak about the use of these agents that we call immunotherapy, sometimes a little too enthusiastically, because it's only kind at the time being two real application, even if I believe in TIGIT, it's still two applications, which is mainly the anti-PD-1, anti-PD-L1, with a growing number of compounds. Remember, no MTD, looking like all of these compounds, maybe apart from the cost, we hope so, will compare similarly or perform similarly in our patients and across diseases. So these are really kind of encore drugs, if you allow me to tell that. But just wanted to stress with you that there is also this growing group of CTLA-4 inhibitors, which are, for most of them, differently engineered. So and they have an MTD, they have really variability in their activity and efficacy on the contrary of anti-PD-1, anti-PD-L1. So what I will tell today about CTLA-4 is speaking about tremelimumab and ipilimumab, but it might be that this experience empirically over time might be modified, improved, or worsened by the use of new CTLA-4 because they do perform differently from the old ones. And of course, always thinking about the controversy of CTLA-4 to start, but also PD-1, PD-L1, and in general, the immunogenic part of the tumor immune system interaction. This is about the Jim Allison slide, always telling you that we still are understanding what we are doing when we handle an immune component, particularly looking at CTLA-4, where I will speak about it at the end. There is a controversy about how it works, still remaining in the community of immunologists. But basically, what we discuss today is not the high PD-L1 that Martin will discuss later on. It's not the chemo IO backbone, which will just be the comparator in my talk today, but it's really the last square of the guidelines, which is these trials, Checkmate 227, Checkmate 9LA, and Poseidon, which have defined potentially the question of CTLA-4 usage. And this is, I would say, the scheme of my talk. The idea is to try to imagine why and if CTLA-4 could overcome biomarkers-defined immune resistance, and basically the main part is PD-L1 negativity, but maybe also the STK11 KEAP1, and just a second about brain mets. So first, but not last, we were asked, by the way, in the past, in the recent weeks, by the GCO to write a summary of what we are with CTLA-4, and we did it with Jordi Ramon and the colleagues, in order just to summarize what I will tell today, so everything is in a table in the GCO. So basically, why do we believe in CTLA-4 in every disease, across diseases? Because we come from melanoma, like everything in oncology, and we had in this initial trial comparing Nivo, Ipi, and Nivo, this feeling that long-term benefit, which is now probably cured in melanoma, was not strongly better, but slightly better when the CTLA-4 was added to the component of nivolumab. So ipi-nivo was better than nivo, even if it was not the aim of this trial to compare, this was the insight we had. And this has been supported by many, many data, mainly done by immunologists or translational immunologists, showing that it might be about T-cell repertoire enlargement. When you give a CTLA-4, you will be able to recognize more different neoantigen by new T-cell repertoire clones, as well as create specific memory T-cell clones. So there is a rationale for the belief. And there is a rationale not to believe that anti-PD-1, anti-PD-L1 would be sufficient to maintain long-term benefit, and this is unfortunately the long-term result presented by Marina and Sylvia, showing that if you look at long-term Keynote 189, Keynote 047, you will agree with me that being progression-free at 5 years is a miracle in squamous and in non-squamous. So we can't do it without a CTLA-4, that is for sure. And this is a Suresh Gadgil wonderful presentation of the pool of the PD-L1 negative patients in these keynote trials in lung cancer, really showing you again how this curves crash at 5 years, meaning that none of these patients are long-term benefiting from an anti-PD-1 plus chemo or without chemo, really meaning that these PD-L1 negative patients are probably the worst to try to imagine being treated with this strategy. And then we can go through the supporting data, showing you that we need additional, not that the data are not good, but we need better proposals to our patients. I like this trial. It's not just an example because it's all histologies. So the Empower Lung Tree had the benefit of it being extremely well designed and having all histologies stratified, so you can look at the whole picture. And remember that adding cemiplimab plus chemotherapy has led to a very significant benefit in overall survival, hazard ratio 0.7 plus. So it's really something which has changed our paradigm. But just to show it's not enough, unfortunately, cemiplimab for this 25% of patients PD-L1 negative is really when you look at this level of PD-L1, the benefit is really stringent, but of course in the underpowered subgroup of negative PD-L1, it's very difficult to decipher a benefit. This was not the question of the trial, but basically it's very difficult to decipher a benefit. And this led, as the usual EMA, non-approval of the strategy in negative PD-L1. That's an habitude, a habit now in EMA, and they did it for this trial not to encompass the negative PD-L1 in the potential registration, and this was the case, as you can see here. Okay? So basically, that's the background of what I will discuss. I'm not sure in this crowd I need to explain, but the 227, basically ipi-nivo versus chemo, in two almost separated trials, positive or negative PD-L1, very last trial in negative PD-L1, by the way. This 9LA, same thing, nivo, EP, low-dose EP, but 2 cycles of chemo added versus chemo. This is cleaner trial, no change in the endpoint, well stratified, OS at the primary endpoint. And the Poseidon, which has the charm of having an arm without the CTLA-4, is randomizing this patient to durva-tremi chemo versus durva chemo versus chemo. So duration, immuno is 2 years for the ipi-nivo, and durva-tremi, to know, is forever until progression, just to show a bit the difference. Okay? So when you look at, first of all, let's start with 227. This is the updated data from last year that I had the pleasure to present. What you can really see is suddenly you recover this subgroup of negative PD-L1 to extract the similar benefit from the strategy of immune addition as compared to the positive. The trial was designed on that subgroup, but the long-term numerical benefit, as well as to even a more important extent, the hazard ratio, will read very positively in negative PD-L1. If the hazard ratio reads better, it's because chemo is really doing worse in negative PD-L1. But basically, look at the number at 6 years. They do the same in negative and positive. So you have recovered an immune response thanks to the CTLA-4 in this patient population, which had not before. Same thing for the duration of response. You recover your duration of response. Marginal trial will tell me, and 227 has never led to any registration, at least in Europe, but you can do the same exercise through the 3 trials. So this is the 9LA, and we will have soon an update. I'm looking forward because I'm sure the later reading is better. The inflection point is after 3 years usually, so I'm looking forward to see a later time point of that trial. But anyway, so 9LA, if you look, this is a positive PD-L1 subgroup. This is a negative PD-L1 subgroup. So again, not only that you can see the benefit, but I find that the plateauing is perfectly interesting here and really making us looking forward for the next update. So 9LA, same feature, you recover the negative PD-L1. And last but not least, the charm of it, the Poseidon has the two. So one without tremelimumab, without CTLA-4, and was with CTLA-4. And look at the last line of my red square, hazard ratio 0.98, you add a CTLA-4, 0.81. And here you can see it with the curves, is where you isolate the clear blue curve as compared to the other ones. So I think there's really no discrepancy to tell about this subgroup. I think it's some years I tell about it, but now I think we are all convinced, so I'm very happy with that. Long-term outcome, 5 years. So basically, just to tell what we sometimes decide to do or not, you double the long-term survivors by adding a CTLA-4 in these patients who are negative in terms of PD-L1 expression on tumor cells, from 10 to 20%. So this is something we consider as valid as a difference for many things, and I think we should keep it in mind. So maybe just another layer, because we always have this idea of why is CTLA-4. Sometimes you have double feature, and the double feature you might have is STK11 plus PD-L1 negative, because very often STK11 tumors mutant tumors are PD-L1 low or negative. But you can also have it alone. Using a CTLA-4 looks like you can recover not a higher, but an immune response in this patient with STK11. Remember John Haymack and Scullidi's data across all the chemo IO trials. There was no benefit, for example, of Pembro in real world and in trials. And here really with the CTLA-4 you can recover a hazard ratio of 0.7 plus 7.8. So really showing that this subgroup with or without a KRAS is benefiting from the CTLA-4 component. And where I find it interesting is because it's again reproducible in the trials. This Poseidon, where you can see the STK11 having, I would say, an incremental of benefit having a CTLA-4 with the largest number of patients responding, the deepest response, and the longest duration of response when you add a CTLA-4. So the 3 parameters. So again, a reproduced data set, and last but not least, 2 to 7, same thing, STK11 is recovered. So PD-L1, STK11, or both, because sometimes it is both. Enough for STK11? Maybe yes, maybe not. I would say yes. Some say no. And there is a prospective trial, randomized. They call it a phase 3-4, which is looking at STK11 plus minus KRAS plus minus KRAS subgroups, and they compare exactly what we say, the addition or not. So the Poseidon regimen versus the 189. I say 189 because usually these alterations are found in non-squamous. Okay? So this has a primary endpoint overall survival. So very interesting trial, and I think it's fair to do the randomized. It's a large proportion of patients. Last but not least, going back to melanoma, Georgina Long, presenting some years ago this idea that EP is so much better than nivolumab in controlling patients with brain mets. This trial, this ABC trial, is in patients with asymptomatic brain mets, but some had very large lesions, right? And you can see how much the intracranial progression-free survival is better with nivo-EP as compared to nivo. So is it true across diseases, and is it because of the microenvironment modification you could bring in the brain? I don't know, but we have data of EP-nivo in brain metastatic disease, and the feeling we have is not only that the efficacy is maintained despite the presence of brain lesion, because you might tell it for any immune strategy. But as compared to the chemo control arm, there is really an incremental magnitude of benefit in this patient with brain mets, really meaning that whatever you do with the usual treatment, you do something more by adding a CTLA-4. You can see you improve the hazard ratio, and really you improve the numerical long-term outcome in the patients with brain mets, regardless of the PD-L1 expression, right? Same thing for 9LA, where you could really see that the patient with brain met had that ratio 0.43. All these brain mets were treated and asymptomatic, important to say. Without brain mets, 0.79. So really something about modifying the evolution of brain mets by using a CTLA-4. I like this intracranial PFS in 9LA, which for me suggests that there might be a role of CTLA-4 in this specific subset. This is ongoing in 3 randomized phase 2 trials, and we need, of course, to have more prospective data. Just in the 30 seconds I still have, new CTLA-4, I wanted to think about, of course, this MEDI 5752, which now is, you know it, a PD-1 CTLA-4 bispecific antibody has been shown to be better blocker of both PD-1 and CTLA-4s than giving both compounds together. This is about the phase 2 trial, where it was better to give it with chemo than pembro with chemo. And very importantly, I didn't put the slide, but this is now in phase 3 trial to prove the concept. So it might be the new CTLA-4 will be better. It's enrolling, currently enrolling, and very successfully enrolling with stratification, of course, by PD-L1 and endpoints defined in negative PD-L1. And two other anti-CTLA-4. One is the one we speak about since a while, the BMS one, the non-fucolizated or xylated CTLA-4, which by the modification of this fuccolization is allowing to reduce the toxicity. So to hide this efficacy outside of the cancer cell microenvironment. So reducing the toxicity, it is in phase 1 in solid tumors. But I like the other one, right? This compound, which is moving from one pharma to the other one, so I can't tell you which one where it will really be developed. But this is the BNT316, which is a compound which has an engineered Fc component with, and I go back to my controversy in the beginning, is allowing to be an active component of Tregs regulation. So the Fc component is perfectly activated in order to give rise to an ADCC, an antibody-directed cell cytotoxicity, and it is thought that Tregs are strongly expressing CTLA-4. So by doing that, they act, of course, of all the priming phase of the immune response, but the idea is to probably also act in completely eradicating the Tregs component in order to magnify the benefit of this CTLA-4. So this is now in phase 2 trial, but look at the phase 1 trial. All these patients are, most of them have been, I think all of them, by the way, are ICI resistant, 27 patients. And as compared to all we have been seeing recently, the response rate was 30%. I like the waterfall plot. So there's really something in this component. Remember the TIM3 PD-1 with 5% response rate. Here we have 30% response rate. So this is going in phase 2 now, probably in phase 3 quite fast, to try to see if this preserve strategy might be a one to move forward, at least at resistance or even frontline. So a very interesting new compound. I say it's probably now we don't really know who will develop it massively, but it's a wonderful compound. So that's it. So are you convinced? For whom would you use it? Fragile, toxicity, I perfectly know that the patient subgroup is defined, but we really have the question of learning curve, of management of toxicity. Is the toxicity prohibitive to you? And do you need, what prospective trials do you need to target negative PD-L1, maybe STK11 keep one, and maybe brain mets? And I think that are the questions we have to have in each of our practice, I would say, cases. And with this, I thank you for your attention.
No, at the end. Thank you, Solange, for this comprehensive review. Next step, we are still waiting for Dr. Reck in there, so we move to the Dr. Delvis, dealing with the newcomers in the combined immunotherapy. Delvis?
Thank you, thank you. Thank you again to everyone to be here at this time. And thank you to me also. I'm going to speak about the combo, which one is the best combination to win the chemo plus anti-PD-1 or anti-PD-L1. I'm not going to speak about the combo monotherapy, you know, anti-TIGIT plus anti-PD-1 or anti-LAG3 anti-PD-1, because I suppose that Martin Reck is going to speak about that. These are my disclosure. We have gained a lot of things with immunotherapy, anti-PD-1, anti-PD-1 over chemotherapy alone, and we know already that. But really, if you see here, in this part of the curve, we have a lot of patients in the first years in all, in the KEYNOTE 189, in the KEYNOTE 407, in all of them, in the first year, around 30 or 40% of the patients are died. And it's clearly almost in the first 6 months, primary resistant. But then, even if we get a gain in overall survival in the end of the curve, you can see that the secondary resistance is an important problem up to now, and we need to improve this. But how can we break the limit of lung cancer survival? Or we can say that, you know, in this meeting we have, how can we redefine the limit of lung cancer survival? Well, what do we need to add on top of anti-PD-1 and anti-PD-L1? Well, we have tried a lot of combination. The first one that was presented in last December was lenvatinib over chemo, and it was the LEAC-06. It was the chemo 189 plus lenvatinib. I have to say that I suppose to be a good result with this, but I have to recognize the LEAC-07 was negative, in strong positive, but not only in first line. In second line, the LEAC-08, you know, was negative also, unfortunately. So, in my opinion, it's just simplistic to think that just adding one drug to the standard is enough to beat the result that we have gained with chemo plus anti-PD-1, anti-PD-1. Anyway, how to overcome this resistance? Just adding a co-stimulatory or co-inhibitory receptor, as I said, is quite simplistic because I think we need to individualize. But stay tuned because tomorrow Jaruska is going to speak about this. Okay, well, in our fact, we have a lot of anti-TIGIT right now in this race in immunotherapy. Since the first was discovered, the TIGIT gene in 2009, right now we have a lot of them. Some of them with FC region active, but others with FC region inactive. And we can speak about that because it is important. It is so important. Solange was talking about this with CTLA-4, and we know that with CTLA-4, the level of Treg, those anti-CTLA-4 with FC active can deplete the amount of Tregs in the microenvironment, and it could be part of the good result of these drugs. But maybe with those anti-TIGIT with FC active, even not only with Treg, but also with the myeloid suppressed cell, is an important issue. But we can discuss later about this because right now we don't have exactly the truth. But I should say that this presentation is going to be like those sessions in ASCO tips, trial in progress. Because unfortunately, if we have a lot of trial with anti-TIGIT and chemotherapy, even in the adjuvant setting or after chemoradiotherapy, we don't have any one positive trial yet with chemo plus immunotherapy. Well, but in the other hand, we can say also that anti-TIGIT, if you focus only in the CD155 on the ligand C226, is so simplistic also, it's so simple, because there is other compound, PVRIG, or another that play an important role in this part. Well, I'm sure you remember the result of CITYSCAPE. Was a phase 2 atezolizumab plus tiragolumab versus atezolizumab was positive. All of us was encouraged, thinking that this combination, maybe in the strong positive population, could be a good combination, especially for a chemo-free combo. But unfortunately, and also we have the ART7, the randomized trial, is also a phase 2, remember, with a small number, like CITYSCAPE, but in this case was domvanalimab, zimberelimab. It was positive. But if you see the number in the strong positive population, it is positive, but also the response rate, the median at 1 year, is not so impressive. So, but I let this for Martin Recht after that. So, unfortunately, we have a press release from this trial, the skyscraper one, who said that it's not positive. Maybe tiragolumab plus atezolizumab, even in that population with PD-L1, more than 50% is not the standard of care. I have to say that in the CITYSCAPE was also a biomarker study taking into account the PVR receptor, and even in those patients with PVR positive, the trial was not different between those arms. So, but we have a lot of trials, as I said, TIPS trial in progress. We know that the skyscraper 6 is like the chemo 189 versus chemo plus atezo plus tiragolumab in non-squamous metastatic non-small cell lung cancer. It was closed in August 2023. We're waiting for this result, but up to now, we don't have even, because it was a phase 2 and then phase 3, we know that the toxicity is not worse than the toxicity with chemo plus immunotherapy, and the data monitor committee allowed to continue with the trial, but we don't have the trial published with the combination, neither in the phase line with chemo plus atezo plus tiragolumab. So we need to wait. I have to say that I read that in squamous esophagus carcinoma, there is one positive trial with chemo plus tira plus atezo, but the control arm was just only chemotherapy. We need to wait. Well, we have also another combo, vibostolimab, pembrolizumab, plus chemotherapy. We have the phase 1 data with the combo, not with chemotherapy, but only vibostolimab plus pembrolizumab in patients previously treated with anti-PD-1, anti-PD-L1. Look at this, the response rate was just 3%. We saw last December in the ESMO, the result of the KEYBITE-002, it was negative. I think the rationale was very soft to do this phase 2 trial. And then we have the phase 1 vibostolimab plus pembro, but we don't have the phase 1 published vibostolimab plus pembro plus chemotherapy in those patients naive for chemotherapy or for IO, and in those patients, in all-comer population, the response rate was 26%, and in those patients with PD-L1 positive was also 33. It was modest also, this result in this population. And we have also a phase 3, the KEYBITE-007. It was compare chemo plus pembro plus vibostolimab in squamous and in non-squamous, and this trial finished in April last year, and we are waiting also for the results of this trial. And we have another combination. We have the combination of ociperlimab, tislelizumab. But in this trial, we have the data of this first phase 1 trial comparing ociperlimab, tislelizumab plus chemotherapy in squamous and non-squamous histology. And in squamous, the response rate was 57%, and in non-squamous, 54%. It's positive. But if you remember, in squamous with the Keynote 407, the response rate is around 60%. It's not too much. The good thing is that the toxicity is not bad with adding an anti-TIGIT, but we need to see a positive result. We have another phase 2. It is an Advantage 105, the first one, and an Advantage 205 is the first 2, 3 that we are waiting also for the result. And we have another phase 3 trial running right now, that is the STAR 121, and with other combination of anti-TIGIT, anti-PD-1, in this case, domvanalimab plus zimberelimab plus chemotherapy, and this trial is running right now, and we need to wait to see the result. But look at how many trials we have with chemotherapy plus anti-PD-1 plus anti-PD-L1 plus anti-TIGIT, without anyone positive. But if you do a research in clinical trial, you will find a lot of them also in the neoadjuvant setting, but also after radiotherapy. It's just to summarize here, the top one is just the combo, the chemo-free regimen that maybe Martin Reng will speak about this. And in this part, all the trial that we have right now, the skyscraper 06, KBI 07, the STAR 121, and the Buddy 205, and we are waiting the result to see if finally it can beat the standard of care of chemo plus anti-PD-1 or anti-PD-L1. A lot of anti-LAG3 also in trial, we have a lot of them also since the beginning. The mechanism of action is a little bit different, and it's more related, this co-inhibitory receptor is a little bit different from the anti-TG. We have a lot of them right now running also, and we have another kind of bispecific also, anti-LAG3 and anti-PD-1 or anti-PD-L1. We have this combo with relatlimab and nivolumab approved by the FDA based on progression-free survival in melanoma. We know that LAG3 and TIGIT are co-expressed with PD-1, and maybe there is a rationale. Yes, there is, but we need the result. And we have also the relativity 104 that it was closed in June last year with a combination of chemo plus nivolumab plus relatlimab. But look at the control arm. Which one is the control arm? Chemo plus nivolumab. Is it really the standard of care? Chemo plus nivolumab? Maybe chemo plus nivo-ipi, maybe in the HMA-NILA. But just plus nivo alone, at least in my opinion, this control arm is not the best one. We have another anti-LAG3, but it's different in this case. This is already, in fact, it's not an anti-LAG3 because the mechanism of action is to activate the dendritic cell. Is a little bit different, but we have this data with a combination of FT plus pembrolizumab in patient naive, and in the strong positive population, it's not bad, but we need a phase 3 to confirm this. We have a lot of trials, but even we have more data with the combo with relatlimab plus nivolumab in the adjuvant setting, but not with chemotherapy. It's just the combo anti-LAG3 plus anti-PD-1. And we have another one, fianlimab plus semiplimab plus chemotherapy that is running right now, and we have to wait to see also the result, but it looks a very good combination also. It was from ChatGPT I did yesterday, and neither ChatGPT know which one is going to be the best in this immunotherapy rate. Just as a summary, I have to say that up to now we have a lot of promises, but really few certainty up to now. We have a lot of promising phase 1 and phase 2 trial, but we have a lot of phase 3 pending or negative up to now. But I always said, do we really need 20 anti-PD-L1 or 20 anti-LAG3, anti-TG? I'm sure that we need better translational, better personalized trial with immunotherapy, because we are losing a lot of patients in this immunotherapy rate. Thank you very much.
Thank you very much. Martin Reck didn't arrive, right? So we continue to video of recording of Dr. Garasino, previously from Milano, currently from University of Chicago, that will discuss the fascinating issue of ADC's combination in first line.
It should be a face-to-face meeting. Fortunately, last time some speaker have to send the presentation, but well, anyway. It's always nice to see Marina even in the video.
Hello everyone, my name is Marina Garassino, and I am really happy to be here with you today, also virtually. I just realized in this moment that Zoom removed the possibility to record the presentations, so I'm using a different app which is called Loom, and I really hope that you will receive my file. So the title of my presentation will be the anti-PD-1, anti-PD-L1 plus the antibody-drug conjugates, the new standard of care in first-line non-small cell lung cancer. Here you have my disclosures. So just to start, I will talk more about the TROP2 compared to other targets, because TROP2 is the target that is in the last until 2024 is the most investigated antibody target in the space of antibody drug conjugates. TROP2, just to remember, is a transmembrane glycoprotein that is overexpressed in many solid tumors, including breast cancer and non-small cell lung cancer. And it is targeted with the antibody drug conjugates with one part, which is a monoclonal anti-TROP2 monoclonal antibody. There is a cleavable linker, and then there is a payload. The payload is the part that is mostly different among all the antibody drug conjugates. Starting from dapotoma deruxtecan is the first TROP2 targeted antibody drug conjugate, was investigated already in phase 1, phase 2, and also phase 3 clinical trials. This is the first trial that was a pan-tumor trial, enrolling also a cohort of non-small cell lung cancer. And as you can see in the dose expansion, there were at least 3 doses, and the 8 milligrams per kilogram every 3 weeks was established as a MTD. However, the 6 milligram per kilogram every 3 weeks was chosen for the development. As you can see here, there are the first results. There is an overall response rate around 22-26% with the different drugs. And as I told you, 6 milligrams per kilogram was chosen as for the future. And you can see that the median progression-free survival of this drug in a cohort, highly pretreated cohort of patients, was about 7 months. We have also results not just for patients without actionable alterations, but there are also results for patients with actionable genomic alteration. And as you can see that also in this case, independently by the mechanism of resistance or by the alteration, the majority of the patients responded to the drug. I'm sure that you remember that this trial, the trial of Dato-DXd over docetaxel in the second line, was presented in the plenary session in ESMO some months ago. And this was a randomized clinical trial comparing Dato-DXd over docetaxel, and results showed that there was a benefit in terms of progression-free survival corresponding to an hazard ratio of 0.75 and a median progression-free survival of 4.4 months. However, you have to remember that if we look at the subgroup analysis, it seems that there is a clear detrimental effect for the squamous population compared to the non-squamous population, suggesting that for this drug, the squamous population is not the right population to develop the drug. Results for the overall survival are very early, but median overall survival was about a year, but there was no benefit over docetaxel, although you can see that the majority of the patients are censored and the results were very early. But it is also important to remember that the hazard ratio for the squamous population was 1.32, suggesting a detrimental effect in this population. This is the TROPION-Lung2, that is Dato-DXd plus pembrolizumab plus or minus chemotherapy. So all these drugs are now moving to the investigations in the first line, which is the topic of my presentation. And you can see that in particular, there are 2 cohorts, one combining Dato-DXd with pembro, and the second cohort combining Dato-DXd also with with carboplatin. As you can see here, you have the results. On the left-hand side, you have the results for all patients, for the doublet and for the triplet. And on the right, you have the results of the patients in the first line, where you can see that there is a 50% of response rate in the doublet, so without carboplatin, and there is a 55% of response rate for those patients who received the triplet. In the first line, however, we don't have the results for the progression-free survival yet. What I think is also very important to mention is that clearly this is paid with some toxicity, and we have to remember that whatever regimen you use in the first line, the toxicity is sometimes not that much, in particular if you use a pemetrexed regimen, but when you combine with the antibody drug conjugate, in particular this antibody drug conjugate, you can see that there is nausea, stomatitis, a decrease of the appetite, and then there are anemia, constipation, and there is also some bone marrow toxicity. And in particular, is also important to remember that there is a class effect of toxicity for these compounds, which is the ILD, but in this case, you can see that the ILD adjudicated as drug-related are very, very few for this particular drug. This is the Tropion-LAN4 that basically is a very similar trial, but instead of using Pembro, used durvalumab plus or minus carboplatin. You can see that there was also a sequential dose escalation, and there was also a dose expansion. The endpoint of this trial was the safety and the tolerability and the possibility, therefore, to combine the Dato-DXd plus the antibody drug conjugate, in this case, durvalumab. Here you have the main baseline characteristics that you can see are very similar for the doublet and also for the triplet. And at the time when the trial was conceived, the squamous population was included. And as you can see, there are in almost 25-30% of patients had a squamous histology. Again, the safety, as I told, was the primary endpoint of this trial, and you can see that the toxicity for the combination with durvalumab and chemo is important, and in particular for the triplet, we have a grade 3 or more treatment-related adverse events in about 70% of the patients compared to 40% for the doublet. And again, in the trial, there were some febrile neutropenia, there were some ILD, and all the side effects that we saw also in the previous trial. And you can see here in bars. So there is the stomatitis, there is the bone marrow toxicity, and there is also some nausea. The preliminary results suggested that the response rate was 50% for the doublet, and it was 76.9% for the triplet. However, as you can see, the results that are available now are not that many. There are many trials that are ongoing, and some of them are in the metastatic space, in particular the Tropion-LAN7 and the Tropion-LAN8 in the patients without the antibody, without the actionable genomic alteration, and some others are for those patients with the actionable genomic alteration. And of note, there are also trials that are exploring the possibility to combine osimertinib plus the Dato-DXd. The second drug that I believe is very important for this presentation is called sacituzumab govitecan. This is exactly another you can see that there is one part which is an antibody, a monoclonal antibody targeting TROP2. There is a linker, and there is a payload. In this case, the payload is called govitecan. The drug is already approved for at least breast cancer and urothelial cancer in the last couple of years, and now it is under investigation for non-small cell lung cancer. The results of the preliminary results that we have for the efficacy of sacituzumab govitecan show the response rate of 16.7% with a progression-free survival of 4.4, which is very similar to the response rate that we observed also for the Dato-DXd. Despite we are targeting exactly the same target, which is the TROP2, I think that the linker and the payload, maybe they have some importance, because as you can observe, in this case, the stomatitis that was present in the other drug is less present in this drug. There are some nausea and some diarrhea, and in particular, I think that nausea and diarrhea are the most relevant toxicity for this drug. And again, you can observe that also in this case, there can be some leukopenia. This is the EVOCE-2 trial that is a phase 2 trial exploring the combination of sacituzumab govitecan and Pembro plus or minus the chemotherapy. So very similar to what we observed in the previous case. And here is the study design. So there is one cohort for patients PD-L1 more than 50, one cohort for patients PD-L1 less than 50, and then there is a safety run-in cohort for patients combining combining also carboplatin or cisplatin. This is a global trial that is supported by Gilead, and we have also some results of the first cohort in terms of efficacy. As you can see here, you have the main baseline characteristics that are very similar for the patients with PD-L1 more than 50 and PD-L1 less than 50%. There is, for the combination of patients with less than 50%, about 50 and 50 had 149 and PD-L1 negative, and a similar number of patients received had non-squamous or squamous histology. Looking at the preliminary results for the efficacy, you can observe that the response rate is for PD-L1 more than 50% is 69%. And I just want to remind you that the average of the response of Pembro alone in this population is about 45%. However, we have also similar results when we combine with the chemotherapy, and we have 44% for the population less than 50%. So the results are promising. It is possible that the activity is increasing. We don't have mature results for the duration of response, and the median duration of response is not known yet. You can see in terms of waterfall plot the change of the target lesions, and as you can see, you can observe that for the target lesions, for all these patients, there is a very good activity, and only very few patients didn't respond to the combination of the drugs. And in this plot, you can observe also that there is some length of response that is exactly what we want to obtain with combination drugs. Also, in this case, this is clearly paid with some treatment-related adverse events. And however, the treatment-related adverse events for the combination of sacituzumab and Pembro are 70% grade 3, so similar to what we have seen also for the other drug, and only few of them, only almost 15% of them were treatment-related adverse events leading to treatment discontinuation. Of note is, again, as we saw before, the diarrhea is the most relevant toxicity for this drug. There is also an important anemia. Only 3 and 6% are grade 3, but as the same consideration that I do also for the Dato-DXd, it's not easy for a patient who potentially can have a good prognosis to live either with the mucositis or with the diarrhea. Reasons for the why we believe that the diarrhea and the mucositis are so frequent side effects are the fact that it's possible that the TROP2 can be also expressed on also in the mucus. Trials are continuing in this space, given the results in particular in terms of response rate. And in particular, I just want to mention that there is the EVOC1, there is a press release that sacituzumab over docetaxel was a negative trial in the press release, and hopefully we will see soon the results, and we will see also soon the results of the trial EVOC3, where we combine sacituzumab govitecan and Pembro over Pembro. These are the two most advanced antibody-drug conjugates targeting TROP2, but there is another one that is called SKB264 that is under investigation of Merck, and you can see here there are good results, at least for the population of patients with EGFR mutants after the response. Summarizing, we have toxicity for sacituzumab govitecan and also Dato-DXd. Remember, for on one side the stomatitis and the dry eye is the most important, and remember also the ILD. On the other side, remember that the diarrhea can be very challenging as well as the nausea. There are also trials that are investigating the use of magic mouthwashes with steroids to see if after 8 weeks we can reduce the incidence and the severity of mucositis or stomatitis. So to conclude, I think that there is a great appetite to move the antibody-drug conjugates in the first line. The activity of the combination is for sure very encouraging. However, the toxicity is discouraging. Mucositis, diarrhea, eye dry, ILD, and I think it will be not so easy to combine these drugs with the it will be not so easy to combine these drugs with the immune checkpoint inhibitors. I would like to thank you so much for your attention. I would like to tell Delvis that I'm really sorry not to be there, also because I promised, but he knows that I had some problems, and I really hope that next year will be the right year to come in presence, and I really hope that this presentation will join because I used a new method that is called Loom. Bye-bye, and enjoy the Congress, the sea, and the beautiful island.
So Dr. Reich is still not arrived, so we move to the discussion. Please, Solange and Dr. Virginia Calvo and Dr. Adel, join us for the panel discussion after the music ends. Because it's music. We have only 5 minutes. Any question from the floor?
I have a question.
Yes.
So to you, Solange, regarding the adding anti-CTLA-4, so there's really good data to support using that for PD-L1 negative. But are you aware of any biomarkers of positively selecting people that would respond to this drug? Like Treg infiltration, for example?
It's an interesting question. For me, being PD-L1 negative is a biomarker, even in its absence. It's a very, very large population at the end of our patients, 1 out of 4. But beyond that, the short answer is no. But the longest answer is, remember the TMB. There's something about to develop a larger T-cell repertoire, which is the basis of the function of the CTLA-4. It might be that you need to have enough potential neoantigen to match your HLA composition to create the T-cell repertoire. And there were quite strong data in MYSTIC, remember that, and in 227 showing that as much as we can quantify, evaluate, or try to have some quantification of the potential neoantigen load at the time being by TMB, it might also be somehow predicting the activity of EP-Nivo, the main of EP, of CTLA-4. It makes sense biologically, but based on all the inequalities, and I would say inhomogeneity we had in trying to quantify TMB, how do you filter, what do you qualify as being a mutation, and so on, we were never able to reproduce it. But sometimes, some people, when they hesitate, they see this high TMB, they still consider that it might be a weak component of the positive biomarker. The question is, why wouldn't AstraZeneca and BMS start to invest in these subgroups? Because we are all convinced about IO. It's because both of the drugs are going out of patent. So building a trial which will read beyond the patent validity, nobody will do. So the new CTLA-4 will probably revisit, I think, from the volrustomig and the MEDI5752 to the other ones, we'll probably revisit these questions of positive selection. And maybe we've gone through TMB, now it might be neoantigen, and we can match and predict better and better. So I hope this will be revisited with the new CTLA-4.
And maybe Treg infiltration.
Yeah, it can be the TILs because you need the TILs, right? Or at least you need them to spark the margin of the tumor, and maybe the brain mets, because going to a phase 3 in brain metastatic patients is something that you can always imagine and consider in our patients.
And what do you think about histology or the smoking habit to choose the patients?
I was thinking to Ralph that you have to give a talk very soon about other biomarkers like gender, smoking habit, the kind of things that we hate to revisit because you have to go back to each trial and look at the subgroups. So basically we are struggling with immuno and gender because the first Lancet paper, or JCO Lancet, I guess, was showing that women are more prone to respond to immunotherapy across diseases. It was making sense because then you were telling more autoimmune disease, so a gender story of immune hyperactivation in women. And then we had this data pooling melanoma and lung, difficult because the two diseases were behaving differently. Better in women in melanoma, probably slightly worse in lung. And then you have to go trial by trial. And if you go trial by trial, I would make it short. If you combine chemo and IO, gender doesn't count anymore. There's only one subgroup of patients who is doing worse being women. We're treated with monotherapy, a checkpoint with high PD-L1. And it might be because under this we have other confounders, the multiplicity. So never smoker, some hidden oncogene addiction, but still, if you just do EGFR-ALK and you don't consider smoking habit in your clinic, women with monotherapy checkpoint, you have to pay a little more attention because they really have less benefit from monotherapy. So I know we all struggle with that. And smoking, for sure, smoking is TMB, neoantigen, so that's absolutely reproducible.
Alfredo?
Yes, I want to ask a question to you, Delvis. You
First
just showed...
Alfredo and then...
Yes,
Okay.
to you, Delvis. Just one question, a great presentation, and I shared your concern about all these anti-TIGIT, all these anti-LAG3. Is there any of them that has got your attention and say, oh, maybe this one might be positive? Any specific one, or it's too early to say? And the second question related to this, you showed the melanoma data with the anti-LAG3, and if you look at the data, I think it works better in PD-L1 negative. Are you aware of any subgroup analysis that is going to be performed in this PD-L1 different subgroup?
Well, it's a very good question, but my answer is I don't really know which one is going to be the best. We need to wait the result. But I just really believe we need better biomarker. Which one? I don't know. Because at the beginning, we thought that with anti-TIGIT, maybe the expression PD-L1 was compulsory to have a response. Right now, we don't have 100% clear that also. And if I have to choose, I have that question for so long also, which one is best, those with FC active or inactive? Is because if you say, if it is active, ADCC can produce a deplete, not only Treg, but also the cytotoxic T-cell lymphocyted or NK cells. But at the end, I think, at least in my opinion, the rationale, those is like in anti-TLF4, those antibodies with FC active, just to answer
sure.
The question which one could be better anti-TIGIT, I prefer those with FC active.
Okay.
It's interesting
but
because,
but I don't know.
It depends on which company you are, because GSK and Roche has FC active components of TIGIT. So there's all this scheme from GSK, which is really due, which show that TIGIT is expressed by Tregs. So when you put an antibody with an FC component which is active, you deplete the Tregs. It makes sense. On the other hand, Arcus, as you know, has an FC engineered inactive TIGIT, which in the clinic gives some signal of potentially slightly stronger activity, but it's small numbers. And they also have all the mechanic around NK cells and ADCs that might be the reason why inactive components. So I still think we still struggle with this FC. Remember, we struggled with avelumab in the past. Is it better or not, and so on. So I think we still struggle about the real difference it can make in the clinic and in the translational setting of the clinic. I really don't have a firm answer for that.
Yes, I agree, but we do believe is that we need to personalize immunotherapy. We need to stop to do the same trial for everybody, giving them all for this combo for everybody, because we need to find the mechanism of resistance even with immunotherapy in those ways. I know it's not easy because we don't know how to do it, but we need it.
But as Solange mentioned before, the majority of the biologic biomarkers are not binary, and there are complex, even for PD-L1 expression. So probably we need to mix clinical potential biomarker with immunologic biomarker in multiparametric models, but that need a lot of patience, but could be created combining different trials, at least in my opinion. What do you think about it? We need to mix the different trials in order to define
That's the hope and the hurdle for me of AI, because using AI, looking at multiple parameters, which can be from 10 to 100 or 1,000, AI will always find a solution where a certain signature might give rise to a positive predictive value. Two questions there. Is it true reality? or just because you fish and you have a fish and you are successful. The other question, because you have to make sense. And the other thing is, for me, the most important thing is seeing a little bit, traveling quite a lot with the foundation now, is applicability, right? How can you today use very complex signatures in order to deliver a treatment? And this is maybe possible in the US and in our countries in the future, but this would be almost impossible to do elsewhere. So I'm not so sure that we will really be so much better for predicting PD-1, PD-L1 efficacy in the future. But remember, we've been giving chemo without any biomarkers and without a lot of efficacy. But I'm not sure we really, I think we have the low-hanging fruits, at least for PD-1, PD-L1. But not for TIGIT, for sure.
You have to close.
We can continue with more questions.
We have to finish.
We close the
Yeah,
session.
something's a bit late.
So we close the session. This is now the lunch break. And then I remember to come back on time
When
in
you're speaking in
spite of
Spanish,
the
just to
beach
say
and the
that
sun.